clear? Please confirm in the chat box, is my voice is loud and clear? Can you all hear me? Okay, thank you. Thank you everyone for the confirmation. A very good evening to one and all. I'm Dr. Tanushree, Senior Faculty associated with Biotecnika. Welcome you all to exclusive webinar on regulatory framework for vaccines. Vaccine, not just a word, but a ray of hope to billions of people around the world, is one of the most important discoveries in the history of medicine. These are biological preparations that have been highly successful in preventing infectious diseases, significantly reducing the incidence of childhood diseases and mortality. Special expertise and procedures are needed for their manufacture, control, and regulation. Today, Biotecnika, in association with Biocon Academy, brings you a fascinating webinar to create awareness on vaccine regulation and regulatory affairs. Biocon Academy, a center of excellence for advanced learning in applied biosciences, is a part of Biocon Limited, which concentrates on skill development among the biotech students. In partnership with many reputed international firms and organizations, Biocon Academy aims to provide industry-oriented training programs and skill development program to help candidates secure promising career in the biotech industry. Today, we have with us two proficient personalities in their respective fields, Dr. Ram Gopal and Dr. Om Prakash. We'll be starting this session with the insights from Dr. Ram Gopal, followed by a session from Dr. Om Prakash. Also, there will be a Q&A session at the end. So you can post your questions in the chat box, which will be answered in the Q&A session after his talk. And don't forget, e-certificates will also be provided. So you have to attend the session till the end and you have to submit the feedback form to get the e-certificate. So it's a privilege for me to introduce Dr. Ram Gopal. Dr. Ram Gopal is working as academic manager in Biocon Academy, Biocon Academy Bangalore since 2016, and manages advanced skill development training programs for life science professionals like Biocon KGI certificate program in biosciences and BITS, Biocon certificate program in applied industrial microbiology. Dr. Ram Gopal is a gold medalist in his master's and holds a PhD in plant biotechnology, a professional MBA and postgraduate diploma in patents law to his credit. He has 20 plus years of professional experience in teaching, research, leadership, and managerial roles in biotechnology sector. He has worked on several biotech research projects and have published research papers in peer-reviewed journals. So let's get started. I welcome Dr. Ram Gopal on board. So handing over the controls to you now, sir.
Good evening, everyone. Thank you, Dr. Tanushree, for a nice introduction. I welcome all the students and other professionals to this webinar, and I also welcome Dr. Om Prakash for joining with us for delivering this webinar on vaccine regulatory framework. I will take a couple of minutes to introduce our audience a glimpse of Biocon Academy, which I am proudly associated with for the last 5 years. Biocon Academy, as mentioned by Dr. Tanushree, is a CSR initiative of Biocon, which was started in 2013-14, and for the past 8 years has immensely contributed for delivering the training specific to industry needs, as well as delivering the trained manpower to 40 to 50 different biotech, pharma, and other contract research organizations across India. So let me take a couple of minutes now and make all of you to understand a little more about our objectives, programs, as well as the achievements. As you can see here, Biocon Academy was established as a center for excellence for advanced learning in biosciences covering different facets of biotechnology, biopharmaceuticals, clinical research, quality, and regulatory aspects. And it is playing an important role to generate more and more employable manpower for different biotech industries. So we have a very clear vision and mission to ensure that the industry skills that are required and the skills that are missing in most of the graduates and postgraduates are bridged, and we prepare the students as a finishing school so that they can enter successfully and start their career in industry. We have a four pillar model in terms of delivering our programs, where we use a strong theoretical learning based on our educational partners like Keck Graduate Institute, California, BITS Pilani, Ramaya College of Arts, Science and Commerce, and of late recently we have added JSS Academy of Higher Education and Research. We also have functional visit partners of Biocon, Syngene, and hands-on training partners of Biozine, Thermo Fisher, and Narayana Hrudayalia. We have excellent professional skill training expertise available in-house, as well as we have communications coaches from corporate trainers. So we use this four pillar model to ensure that all the candidates who enroll to Biocon Academy programs get the best world class training and exposure, so that they are prepared for any such pharma biotech company, whether it is a small or medium scale or a large company or an MNC. We have a strong pedagogy where we have a proactive learning model, where we transfer the onus of learning responsibility onto the students by way of conducting project based learning, problem based learning, where we introduce several industry specific case studies so that students actually get to know how they could apply their knowledge and skills in industry when they get into different roles. If you see, we had a successful, eventful milestones all these 7 years where we launched different programs catering to industry needs, starting with the flagship program called Biocon KGI program in biosciences, which so far we have completed 18 batches successfully with 100% placement and trained more than 450 candidates from all over India. We are currently running the 19th batch, and 20th batch will be starting from 21st of October once the placements for 19th batch is over. We also started a program with BITS Pilani from different campuses in applied industrial microbiology, where we have so far completed 7 batches of this quality control microbiology program for the benefit of microbiology and life science postgraduates who want to enter into industry roles for QC microbiology roles. We're going to start the 8th batch from February 2022. In 2015, we started a new initiative to train the in-house employees of Biocon by way of a Biocon certificate program in bioscience management. This was initially offered for managers. We are shortly planning to open a full-fledged MBA for different industry professionals. We started a clinical development program with KGI in 2017 to cater to the B Farm, M Farm, Farm D, and other life science students who want to make their career in clinical research organizations. And we have so far completed 3 batches of this program. In 2017, we also started a new initiative to include the faculty development program so that different life science teaching faculty from universities and colleges get a chance to visit Biocon and Biocon Academy to get an industry update so that they can go back with rich experience of how different subject knowledge is translated in industry by way of different products, so that they can teach effectively, they can improve their curriculum, they can guide the students as well as propagate this industrial skill development program initiative of Biocon Academy. We also started a program with Ramaya Group in 2019 in quality control analytical, specifically to cater to chemistry and biochemistry postgraduates, where they are mainly trained on analytical instrumentation that is associated with quality departments. This year, we are very proud to announce that 30th August, we're going to launch the Global Regulatory Affairs Program in association with JSS Academy of Higher Education and Research, which will be our 8th such flagship program under the auspice of Biocon Academy. I'm also very happy to share that all our programs comes not only with industry oriented skill development as well as training and case studies as well as exposure to the leadership. They are associated with 100% placement opportunities in different biotech and pharma and bioservices industry. This slide tells you a glimpse of some of the companies which have hired from us so far, and they are always happy to come back to Biocon Academy because they have seen the value add that our students actually get by way of getting the industry relevant skill sets. We also are very happy to share that it is not only catering to R&D, production, quality, regulatory departments. Many of our proud alumni are working in varied, diverse roles in life science industries, be it drug safety, marketing, medical writing, project management, and corporate communications, and so on. So we therefore are preparing the candidates for industry mainly with respect to the production R&D and quality departments, but at the same time, depending on specific skill sets, they are also available to be hired by companies for such interdisciplinary and transdisciplinary roles. And I welcome all of you to visit our website, talk to us. You can mail us, you can call us, and see that you actually clear all the queries and doubts associated with your career planning, as well as if you are interested to enter into industry, we are always ready to embrace you. Thank you and all the best for you. And I give back this platform to Dr. Tanushree for taking this forward.
Thank you so much, sir, for bringing more light to this amazing organization. Thank you very much. Now, you'll be hearing Dr. Om Prakash, who is here to enlighten us on regulatory framework for vaccines. It's a great honor and privilege for me to introduce Dr. Om Prakash. Dr. Om Prakash is currently working as the technical director and partner Vumas Consulting. He holds a postgraduate degree in pharmacy, business administration, and a PhD in business management from Aligarh Muslim University. His research thesis was on evaluating and optimizing the influence of business models on the business performance of life sciences organizations. He has 30 plus years of experience in regulatory affairs, pharmaceutical drug development, clinical operations, quality management, and product marketing in many reputed pharma firms. Now, without taking much time, I welcome Dr. Om Prakash on board. Handing over the controls to you now, sir.
Thank you, Dr. Tanushree, for that wonderful introduction. Thank you, Dr. Ram Gopal. And thank you to all the audience who have come in at such a late time in the evening after a tiring day. Hopefully, I'll be able to hold your interest and sort of pick your enthusiasm towards working in the field of regulatory, um, in the field of regulations and regulatory services. Okay, that's what I'm trying to say. Now, just to understand the type of people who are there in this seminar, I was informed that we'll have some students and working professionals also. So can I just ask you to comment in the chat box if you're a student with an S and if you're a working professional with a W. So this will help me get a better understanding of how I could better communicate whatever I'm going to talk about. Can I just ask you to type yes or W? Thank you. So I figured that based on your answers, primarily we have around 80% of students in the seminar with us today. So let me try to sort of modulate this communication to help you understand this whole thing better about the regulatory framework for vaccines. Any questions, please type in the chat box so that my colleagues who are working in the backstage can sort of pick and then let us answer your questions. All right, so let's dive into this. So we are talking about the regulatory framework for vaccines. So I will structure my presentation and my presentation, which into the following 5 categories. Number one, we'll try to understand what regulations are, what a medicine is, how the medicine comes from a lab to a patient, what is the cost and time for this journey, what is the regulatory path of this journey, and also, since this talks about regulations about vaccines, I would be talking a bit about what vaccines are, what are the types, what goes into a vaccine, how do you produce a vaccine, and what exactly is the regulatory journey of the vaccines. It would not be, I would say, beneficial without having a discussion, or rather, without communicating the live examples, specifically about COVID-19 vaccines, which I'm sure all of you know is a life-saving. I would say I would at this point in time, I think for quite a number of people, I would categorize that to be Amruta, right? Uh, as in Amruta in Devaloka. All right. So, how many of you have taken a single dose of vaccine, and how many of you have taken double doses of vaccine? Single dose, please type 1. 2 doses, please type 2. So let's see how many of you are really into this vaccination program, successfully involved in this vaccination program. Okay. So I'll wait for the answer. Please type, and then let me know how many of you are. Excellent. 50. Single dose or having taken vaccinations. Okay. So let's try to understand what is that you have taken, and what all a product, a substance has to go through before it comes to a patient and before the patient can consume it. Okay. This is what we're trying to say. And this journey is called bench to pharmacy. There are multiple terminologies. We talk about lab to market, drug to market, etc., etc., etc. But overall, I think what we need to understand is, from a lab bench, how does it come to the pharmacy? When I say pharmacy, pharmacy is the point from where you consume medicines. Okay, medicines primarily are substances which help you uh, cure, prevent, or diagnose a particular disease. That's a very short form of what a medicine is. Okay. Now, medicines can be of various types. Number one, we had Dr. Tanushree talk about biological. So I'll try to sort of link the different types of medicines. Based on the origin of the medicines, they might be naturally occurring medicines, substances which are naturally occurring, substances which are semi-synthetic, chemicals primarily, or substances which are synthetic, and substances which are biological substances, which can be, or rather, which are used as medicines. Now, what do I mean by biologics? How many of you have heard the term recombinant? I'm sure most of you are from the biotechnology field. So hopefully, I think all of you would have heard the term biotechnology. Recombinant. Okay, so vaccines are biologics, and some recombinant technology products, proteins primarily like insulin, etc., etc., are categorized as biologics. So right now, we will not try to understand the differences between the nuances and the differences between the different category of products. What I want you to understand is the cost which goes into bringing a molecule or bringing a substance from the bench to the pharmacy, or from the bench to the patient. In 2003, it used to cost us around $802 million. At this point in time, the cost is between 1.5 billion US dollars to 2.4 billion US dollars. One product to come to the market. Okay. At this point in time, it takes us around 8 to 10 years to bring it to the market. In 2003, I think it used to take around 10 to 12 years. Interesting fact, there is a product which is discovered and brought to the market from Novartis Gene Therapies, which is called Zolgensma, which contains onasemnogene abiporvovec. So how does that sound? Does it sound like Latin, Greek? I don't know, but yeah, whatever. So it is a rare genetic disorder and costs 18 crore rupees per dose. Yeah, 18 crore rupees per dose. Now, why am I telling you these numbers? Why am I telling you these timelines? This is for you to understand the complete journey which a substance undergoes before it comes to you. Now, let's sort of deep dive into what happens. Okay. Now, the whole process of bringing a product to the market, in this case, bringing a substance, a medicine from the lab to the bench, is all about solving a problem. All right, like almost all things in life, it's all about solving a problem. Now, what is the problem? The problem here is a disease which cannot be cured, or rather, no treatment has been found till date, or there are treatments, but uh, they're not that great. Remdesivir, for example, which is used in treating corona, uh, it's not that great, okay, compared to, let's say, the COVID-19 vaccine. All right. So the problem is that, so we might have something like that product which is not really curing the disease, or we don't have anything to cure the disease. All right. So that is the problem. Now, how do we solve it? In the pharma industry, when I say in the pharma industry, it includes in the medicine industry. Let me put it that way. In the medicine discovery and development industry, there is a structured approach on how we go about solving this problem. Okay. So nothing I would say surprising, because it's a very structured approach and a structured process of how we identify the problem. The problem here is the disease, like I was telling you, and how we go about figuring out the solution to solve this problem. So what is that we do? We primarily form a research team with an objective saying that, guys, you know what? We need to find a substance which is going to cure this disease or prevent this disease or helps in diagnosing this disease. Okay, we're putting the best brains in the organization together and telling them, all right, so do something and find a solution for this. Now these guys are going to identify or find out how the disease is formed and what are the specific areas which we can block, or which we can uh, how we can basically prevent a disease. That's what they're going to figure out. Once you understand how the problem is caused, it's easy to find a solution for it. So once theoretically they have identified, okay, this is the way we are going to stop this disease from uh, occurring in humans, then they try to find out the different kind of substances, the substances which can be natural, semi-synthetic, synthetic chemicals, or biologicals, which will prevent this disease from occurring. So here in the slide, you see the second part component, which is in novel chemical synthesis. It could be a chemical natural product or a biological product. Once they have identified a range of products, they don't start with one substance. They start with around 5,000 to 8,000 different compounds. Okay, to sort of understand out of this 5,000 to 8,000 compounds, which probably could work in treating this disease. Okay, once we have identified that, then we go and test this thing in animals. This stage is called the preclinical studies or preclinical testing. Once we test these identified substances in animals from 5,000 to 8,000, we might have brought it down to, let us say, around 100 or 150 compounds or substances. Once we test this in preclinical animals like rats, mice, rhesus monkeys, guinea pigs, etc., etc., based on the type of molecule and based on the type of disease which we are trying to find a cure for, we take this whole data which we have collected and try to submit it to a regulator here. So who is a regulator? We'll talk more about regulations and regulators subsequently, but just for you to understand, I want to introduce the term regulator here. The regulator is the US FDA in the United States of America. It's EMEA in European Union, and in India, it is CO. Okay, so primarily we take this data, submit it to the regulator. The regulator will do an independent assessment of the data which we have provided, and he or she, or rather a group of people in the regulatory in the regulatory organization will determine that whatever you have tested till now is safe and it can be tested in humans. So post that, we test it in humans. Testing in humans will be of three stages. One is phase 1, phase 2, and phase 3. So we call them clinical trials. After running all those studies, we go back again to the regulator and say, you know what? Okay, we demonstrated that the product what we have identified is safe in animals. Based on that, you gave us an approval to test it in humans. Now we have tested it in humans, we tested it in patients, and this is the data, okay, which we have got from all these studies. Please review it independently. So they are going to review it independently, understand the science behind the product, etc., etc., and then they are going to approve this product for marketing. Some interesting facts: because uh, a drug has to be used in human beings, the responsibility of a regulator is that he or she should ensure a safe and effective product is released into the market, because the safety and efficacy is has to be assessed independently without any bias. And because of the intricate nature of this industry is highly regulated. Okay, number one. So what we primarily look at when we say safe and effective, what is the safe and effective? We primarily look at something called the benefit to risk ratio. The benefit, all of you will agree that the benefit has to be high when we take a substance, when we eat a substance, when we imbibe a substance. The benefit should be very high when compared to the risk. Risk has to be very, very low. That's what they're going to assess. And if the benefit is very high compared to the risk, then they are going to approve the product to be given to the patient. Okay. So this whole process, I'll come back to the question: why does it cost me 1.5 billion or 2.4 billion dollars, and why does it take me 8 to 10 years or 10 to 12 years to sort of get this thing to the patient? It's primarily because this whole process of testing is designed to fail, because the objective has to be that anything which goes through this cycle should demonstrate more benefit when compared to the risk. Okay, that's the reason why it is designed to fail. Now, for example, in 2003 or 2005, the clinical development, the molecules, number of molecules which had gone to clinical development, whatever is being tested in humans is called the clinical development stage. So in 2003 or 2005, you had around 22% of success, whereas in right now, when I'm talking about 2020, the approvals have come down to 12%. So basically, what does it mean? If at all I test 8 molecules, 7 molecules fail. Okay, that's the reason for this high cost, and lot of time which is required for bringing this to the product. Hopefully, this gives you an idea on the journey from lab to market. Now, what I'll try to help you understand is, since we are talking about regulations of vaccines, let's try to understand what vaccines are. A very simple definition of a vaccine is a substance that is that stimulates the production of antibodies in humans or in animals. It works for both, not the same product, but primarily a vaccine is a substance which stimulates the production of antibodies. What do you mean by so why does it, what happens if you stimulate the production of antibodies? It primarily provides immunity against one or several diseases. And okay, is this clear? Type yes. If it's not clear, type no in the chat box. Okay. So Primarily, vaccines are prepared from the causative agent of the disease, its products, or a synthetic substitute. And primarily, what happens is a vaccine acts as an antigen, basically an irritant, which stimulates the production of antibodies in the body. That's what it does. But this thing happens without inducing the disease. Okay, understand this. We use the causative organism, causative virus, modify it in such a way that it does not induce the disease, but it acts as an antigen or as a irritant, or rather as a stimulant, which primarily produces antibodies. More complex definition, it's a biological preparation that provides active acquired immunity to a particular infectious diseases. Okay, just to show that yeah, we come from a science background. Yeah. So a vaccine typically contains an agent that resembles a disease-causing microorganism and is often made from weakened or killed forms of microtoxins, one of the surface proteins. We'll try to understand this subsequently. Okay. Now, what are the different types of immunities which come from consuming a vaccine? One is active immunity, primarily an antigen antibody response. So what does that mean? We expose the body to an antigen and generate adaptive immune response. Okay, but this is long lasting. Passive immunity, basically what we do is we give the antigen to another animal or to other. Oops, I think I did something wrong. Okay, all right. Yeah, sorry. So active immunity is something which you acquire, and passive immunity, what we do is we primarily administer the antigen to an animal, the horses, etc., etc. And the antibodies which are generated in the horse's body are withdrawn, purified, and then given to human beings. So that there is passive immunity. The antibodies are not produced within the host. The human being is not the host. It's produced in some other host animal, and then from there we transfer it. We transfer only the antibody. So that's the difference I need you to understand. That's it. Yeah, each topic what we are talking about takes a lot of time, lot of study to understand, but these are very quick summary points which sort of will help you understand what we are talking about. Okay. So what are the different types of vaccines? Attenuated vaccines, inactivated vaccines, toxoid vaccines, subunit vaccines. So attenuated vaccines are primarily what we do is we take the virus or the bacteria which causes the disease and remove the disease causing component of the bacteria or the virus, and then administer this reduced virulence or less dangerous organisms. We basically reduce the disease forming capability of that virus or bacteria, and then inject it into the human being so that it can act as an antigen. Example of viral vaccines are yellow fever vaccines, measles vaccines, mumps, and rubella vaccines. Bacterial vaccine is the typhoid vaccine. Vaccines for typhoid. Okay. Now inactivated vaccines. Now, so it's just an extension, sort of an extension of this attenuated category of vaccines, in the sense that the vaccine is taken, and the virus or the bacteria is taken and it's inactivated using chemicals, heat, or radiation. So we basically we destroy the virus or the bacteria, and then inject this dead organisms into the body, and they act as antigens, which sort of stimulate the production of antibodies, and they in turn bring about immunity against a disease. Example is polio vaccine, hepatitis A vaccine, rabies vaccine, and influenza vaccines. Now, interesting is this toxoid. Instead of the organisms, what we do is primarily we sort of capture. We sort of purify, identify, isolate, and purify the toxins or the chemicals based on which they bring about a disease, and then we use that particular substance as an antigen to bring about the production of stimulation of antibodies. Okay, so example is the tetanus or the diphtheria vaccine. Subunit vaccine, again, all of them are very similar. They overlap with each other. A small part of an organism is used to bring about generation of the antigen. Now there's something called viral vector vaccines. Now what I want to really focus on is the RNA vaccine, because it is something. So in all this presentation, I'm trying to focus on what is very relevant to the latest pandemic, COVID-19. So RNA vaccine, we have one component, or rather one vaccine, which is based on this RNA technology. So what it basically does, it takes the nucleic acid RNA of the virus or the bacteria and packages it within a vector such as a lipid nanoparticle. Okay, both Pfizer as well as Moderna vaccines are mRNA vaccines. Okay. Now again, we have monovalent and polyvalent. Monovalent is a vaccine which is used to treat a single disease. Polyvalent, which can be pentavalent, tetravalent, etc., to treat more than one disease. Now, this is a very interesting slide which I want you to understand. Okay, normally in any medicine, take a tablet or take an injection of any chemical, semi-synthetic or naturally available substance. You typically have two components in this formulation. Number one is the active ingredient, and the second one is the inactive ingredient. For example, let's take the case of paracetamol tablets. Paracetamol is the active ingredient. Everything else, we use starch, we use diluents, we use whatever, etc., 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 so that you get you see the form of the tablet. Okay, in vaccines, primarily there are two active ingredients. Number one is the antigen itself, and the second one is the immunologic adjuvant. Okay, so primarily what it does is it works synergistically and boosts immune response. So it primarily, so for example, let us say the antigen has a 10x sort of a uh, antibody stimulating power. Okay, I'm just giving an example. If you add an immunologic adjuvant, probably this 10x becomes 12x, or it becomes 15x, or it becomes 20x. Not 20x. 20x is too far fetched. So maybe around 12x or 14x, sort of an increase in the total stimulation of the body to produce antibiotic power of the formulation. Okay, that's one. The other things are primarily stabilizers, preservatives, residual excipients. This is very interesting. Again, residual excipients, because we manufacture virus and bacteria in egg proteins. So any egg protein which is left over after purification, very, 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 very minute quantities in some individuals cause anaphylactic reaction. Okay, anaphylactic reaction is something, allergic reaction, I'll say a very severe form of allergic reaction, very, very rare individuals. Okay, so that's also something which is available, and that's why older generation of vaccines had this, and the latest generation of vaccines primarily don't have it. Okay, now so initially, so now let's deep dive into the vaccines regulatory pathway. All right, so you sort of were exposed to the drug development path, and I'm trying to sort of uh, repeat that, including the regulatory components which go into this journey from lab to bench in this slide. Okay, now the one in green is primarily what happens in the US. The one in blue is the regulatory pathway which happens in Europe, and the one in white, green outline with white background, is the process which happens in India. Okay, now very quickly, the investigational product, the product which is being tested, which you have identified, sort of, you think theoretically, you think it works, so it'll be it has to undergo preclinical studies to determine the safety. Then once the data is done, you submit it to the regulator. We call it pre-IND consultation in the US, and you call it clinical authorization application submission in Europe. Once the application is submitted, then the IND application is submitted. Once it is approved, then you do the clinical studies, phase 1, phase 2, phase 3, and finally, after doing all these three phases of the study, you do something called this complete data is submitted to the regulator, and that application is called the BLA application. Okay, what is BLA? Biological Licensing Approval application. Okay, review and approval. That's what happens. So it's very similar in both the countries. Now in India, we have a bit more, I would say, more rigid process. Now, the moment you have identified the investigational product, you'll have to go to the regulator and get his approval for conducting the preclinical studies. Okay, understand, we don't have that approval stage here and here. Okay, in most cases. Now, in all cases, we'll have to go back to CDSUO, our regulator in India, and ask them, saying that, listen, we have identified this, we need to conduct these kind of clinical studies. Oh, sorry, preclinical studies in animals. Once an approval, you get an approval here, then you can conduct the preclinical studies. Then the data what you collect, you again file a clinical trial application submission to CDSCO. He gives you an approval for conducting the phase 1 clinical study. Post conducting the phase 1 clinical study, you'll have to again go to the regulator to ask for his approval, conduct the phase 2 studies. This data, you go back to the regulator again, get an approval, then do the phase 3, then submit a BLA application. Provided all things, provided we can demonstrate with this whole sum of data that our product is safe and efficacious, then they're going to review it independently and then they're going to approve it. So that's when the product comes to the market. Now, in this, how many of you have heard this term emergency use authorization? If you have used this term, please type Y. If you have not heard about this term, please type N. No. Can I get some response, please? Yes for Y, Y for yes, N for no. Type in a chat box. So, emergency use authorization is a mechanism to facilitate availability and use of medical countermeasures, including vaccines, during public health emergencies, such as what is happening in this pandemic. Okay. Now, what I want you to understand, this is a very important slide. This will help you understand what's happening. This will also help you understand that the products which you have taken are safe and effective and why. Okay. Now, same slide, same way like how it is happening in the US, Europe, and in India. Now, what happens is, primarily, any product which has to be approved will have to go through this complete life cycle. You do before it is given to patients. Okay. Now, what UA does primarily, emergency use authorization does, which has happened across the world for all COVID vaccines, which are safe and efficacious, is this. After doing the phase 1 study, there is an opportunity for the regulator to review the data and give an approval. An approval will be, the approval which is given at this stage is called an emergency use authorization. It can happen either during phase 1 or during phase 2. So what does it mean? EUA primarily means it's a restricted authorization. What does it mean? Restricted authorization. You cannot use it to everybody. You have to use it under a specific, very specific conditions. For example, in our case, it says only through a government vaccinated program you'll have to get it administered, etc., etc., etc. Okay. Why is that? Is only because we don't have anything to treat this disease which is affecting us, it's a pandemic primarily. Okay, it's an emergency where you don't have anything to uh, sort of overcome this disease. All right. And that is why the regulator has taken an approval and said, okay, we are going to give this product, we are going to give the substance only to this set of patients, only to these number of patients, and we will be closely observing and monitoring what's happening. If there is an issue, we're going to stop it immediately. If not, we're going to let it move to the next phase. Then it does not mean emergency use authorization does not mean the product is approved to be sold. No. It is approved to be administered under specific conditions to a specific set of population. Okay, diseased people, patients. Now, subsequent to doing a EUA, the manufacturer will go through this whole process. Now, why do you do a EUA? I can't wait for 8 years to get this thing approved. Okay, I want to have something now. I want to have something within 6 months. I want to have something within 1 year. I have to have something within 1.5 years. That's how important it is to stop death of people and things of that sort. Okay, so this is why, after the regulator considers the strong scientific rationale supported by adequate data, and after determining that there is no alternative available, an independent expert committee will review and authorize for emergency use. Again, I want to emphasize, it is not a marketing approval. Okay, all, whatever, everywhere this is the same thing. We can have the EUA can happen either between phase 1 to phase 2, or after between phase 2 to phase 3. Now, EUA need not happen between phase 3 to phase 4, because this is a standard process. It will take its own time, so it doesn't give you any advantage, because this is where you get a lot of time, you consume a lot of time to demonstrate the efficacy of the product. Okay, this is what happens. This is what I wanted you to understand clearly. Now, vaccines, normal pathway, emergency use pathway, or emergency use approval will happen only in case of pandemics and when there are no available treatment for curing a disease. Okay. Now, you have understood this. Now, let me give you an example so that it sits strongly in your mind. Now, I've taken the case of Covaxin, okay, manufactured by Bharat Biotech. I saw that around 50%, almost 80% of, 50% of you have taken vaccines, at least a single dose. You would have either taken Covid Shield, which is manufactured by Serum Institute, or Covaxin. Off late, I think you would have taken Sputnik 5. I'm not sure how many of you would have taken that. Pfizer has been approved recently. So I wanted to give an example of Covaxin because it's purely manufactured here in India. Everything has been done in India. It's a collaboration between ICMR and Bharat Biotech. The government is strongly behind, Bharat Biotech to help them produce. We have this. So what I'm trying to say is, we have the competency, we have the capability of manufacturing vaccines, good products. That's what I wanted to demonstrate. We are able to demonstrate that. Okay. Now, besides that, now Covaxin, what does it contain? It contains whole virion inactivated coronavirus antigen 6 micrograms. Primarily, it is a subunit. It just contains inactivated coronavirus antigen. It can be, we talk about, we categorize this as an attenuated vaccine. Okay. And it is produced using a vero cell based platform that propagates the virus. Primarily, this vero cell based platform helps in the reproduction of the virus and expresses the viral spike S protein. The subunit of SARS-CoV-2 virus, which is available in this vaccine, is viral spike S protein. Okay, that's the antigen which is available. Now, the adjuvants, like I was talking, we have aluminum hydrogen gel, which acts as an immunity stimulator, sort of. It acts along with the antigen, along with this S protein spike which is available. We have TLR 7/8 agonist. Again, this also acts as an immunity booster. Both aluminum hydroxide gel and TLR 7/8 agonist act as immunity boosters. 2-phenoxy ethanol, it's actually a preservative. Phosphate buffer solution is actually the vehicle under which this vaccine is formulated. Please understand, the actual antigen concentration is only 6 micrograms, and you are administered 0.5 ml per dose. Just understand the dilution factor. Quite a number of you come from biotechnology. Just understand the dilution factor. You have 6 micrograms in 0.5 ml. So much of water goes into it. Okay. Now, how was the safety and efficacy demonstrated? Bharat Biotech tested this product for immunogenicity in mice, rats, and rabbits, for immunogenicity, safety, and protective efficacy in Syrian hamster and non-human primate, rhesus monkeys. Okay. Phase 1 studies, they did it in 375 human participants. Phase 2 studies, they did it in 380 participants. Phase 3, which is ongoing, based on, so the approval for the EUA was given at the end of phase 2. Somewhere during the end of phase 2 is when they gave an approval for Covaxin. That is an EUA, not a market authorization. Okay. Now, phase 3 is ongoing. They're doing the study in 25,800 participants. It's an ongoing. Once they complete this study, then they will take the whole data and then they submit it to the regulator, who will look at the data points, who will look at the whole science behind it, and then give an approval for this product to be marketed in India. Okay, then it will be available in all the stores. Okay, so how many of you feel this whole point of understanding the science behind a product, how it is brought to the market, what all we should do in terms of helping the regulator understand that my product is safe and efficacious, what type of studies we should do, what type of data points we should provide, what type of justifications we have to provide if there is something which we have not yet understood. This is what a regulatory professional does at the end of the day. Okay, so for him or her to be a truly regulatory professional, he or she should be looking at the complete picture. It is not only about filling some form, submitting it to the regulator, and no. It's changed a lot. You will have to understand the science. You have to be the ambassador in front of the regulator to demonstrate that your product is safe and efficacious, and that's the only reason why your regulator should approve this for marketing. Okay, I will use that word. You should be the product ambassador in front of the regulator. That's what it is all about. So I think we are almost at the end of my this thing now. How many of you would be interested in pursuing this as a career? Can I have an yes, if at all you're interested in pursuing this as a career, or no, if you think you have something, some other thing which is of more interest for you? Yes, if you're interested. Either way is fine. I can't dive that much into such a beautiful and passionate topic to understand this. Okay, so I think most of you have said yes, you are interested in pursuing this as a career. Now, what Dr. Ram Gopal is saying in the beginning, he was talking about how Biocon Academy provides the required regulatory professional competency training, and also helps you gain placement. Gain placement in organizations. If this really ignites you to do something, please go ahead. This slide gives you all the details about the program. Dr. Ram Gopal already told you about that, but you have the scan QR code. You could call them, reach out to them, get more information, etc., and then get into this. It's for me. It's very passionate. Because I've done a lot of work in this area, and it's good. All right, I think I'm done. Any questions? We could take it. If you have any questions on either the vaccine, the EAU, the regulatory stuff, or Biocon Academy, I think I'll answer on the technical side. I'll answer, and Dr. Ram Gopal will answer on the Biocon Academy specific questions. Please go ahead.
Thank you so much, sir, for such a wonderful and informative and interactive session. Thank you for enlightening us about vaccine types, examples, vaccine regulation. And I'm sure today most of our talented and passionate participants have been motivated to choose a vaccine regulatory affair as a career option. So thank you so much, sir. So now moving on to Q&A session. So let's see our first question. So our first question is from Vibha. Can people pursuing PhD not apply for this course?
People pursuing PhD either part-time or full-time may not be available to take up this because this regulatory affairs program is a full-time course. So we need those who have completed postgraduate degree so that they are able to spend their full time with the industry experts as well as the trainers, right, who are going to deliver this program. And the purpose is to give the skill sets and give them an employment opportunity at the end of the course. So it's always advisable to complete what you are currently doing, Vibha, so that you would be able to later on take this course.
Okay, thank you, sir. I hope that answers your question, Vibha. Can we see our next question? Okay, so next question is from Ash. A vaccine is produced after many years as a result of many experiments. COVID vaccines are produced within short period of time. So is this actually good for our health?
Excellent question. Excellent question. Now, like I was telling you, it is not approved for marketing. It has only been given a emergency use authorization to control this pandemic. You don't have anything. Something is better than nothing is the term which I would use, not in a very loose way. Okay, I would not use the word something in a very loose way. It has gone through tests. It has demonstrated that it is safety. The last slide, if at all you were to look at my last point in my last slide of presentation, I was telling you how Bharat Biotech was able to demonstrate safety and efficacy by administering it in various preclinical models of animals as well as in human beings. And the phase 3 clinical trial is ongoing even today in 25,800 people. So, yes, it's very good. I have not only me, my whole family has taken both the doses, safe, healthy, non COVID. God's grace. And yes, of course, because of the vaccine which we have taken. Okay. Does that answer your? It's absolutely good for your health. Don't worry about it. Just go and take it. The downside is
So I
very
hope,
expensive. Yeah. Sorry. Yeah.
yeah. So I hope that answers your question, Ash. So let's see our next question. Okay, so next question is from Sushmita. Sir, if we could not do the full-time program now, then would this chance be available in future again to work?
Okay. So this Smita, yes, absolutely. We are going to do this maybe another batch in 2022. So once you finish your current course that you are enrolled to, you can again apply to our next batch of this program. This would be continued because we have seen that regulatory requirement is there for industries, especially for the professionals who are trained on either national or international regulatory affairs. So this will be available maybe once you finish your current program.
Okay, so thank you, sir. I hope that answers your question, Sushmita. Let's see our next question. So next question is from Rishabh. Why vaccine is considered to be a last stage, kind of, to treat a microbial infection like SARS-CoV-2, even after reporting many mutation and breakthrough infection?
So, Rishabh, vaccine is the only treatment available at this point in time. Please understand, it works with your body, it works with your immune system. So, your vaccine basically strengthens, builds your immune system. I don't see any other treatment as, I would say, any other treatment as powerful as a vaccine because it works in line or it works along with the immunity system which is already existing in your body. Okay, what else do you need? And it builds your immunity system for quite some time. What else do you need instead of popping a pill, let's say 3 times a day for 15 days, 1 month, 2 months, 3 months? Don't you think having a jab within whatever specified schedule, and building your own immunity. Your body has that thing not only for today, it has it for a long duration of time. Don't you think that makes it a better, better product when compared to the this thing? I would say vaccine is not the last this thing, it's the first defense in the armamentarium for attacking COVID-19, at least for preventing COVID-19 at this point in time. Treatment, yes, there have to be multiple breakthroughs and things of that sort. But yes, for prevention, no two things about it.
Okay, thank you so much, sir. So I hope that answers your question, Rishabh. So this was our last question. I know there are not many questions, but because of time constraint, won't be possible for us to take all the questions here. So we have come to the end of the session now. I once again express my gratitude to both the eminent speakers, Dr. Ram Gopal, Dr. Om Prakash, sir, for taking out time from their busy schedule and enlightening us. It's a lot of takeaway learning experience. Also, I would like to thank all the participants for attending this session. Participants whose queries haven't been addressed in session, you need not to be get disheartened. You can post your queries, you can post your questions on our website zipmessage.com/biotecnika. Also, you can directly you have the contact details as shared by Dr. Om Prakash sir and Dr. Ram Gopal sir. You can directly contact Biocon Academy, and you can drop your questions in a form of video, text, any inquiry, any feedback you want to give, you can always post. So either you can post on zipmessage.com/biotecnika, so we'll get it answered, your questions will get it answered by Biocon Academy, or you can directly contact Biocon Academy. And also, I would like to tell you about the feedback form. So feedback link will be shared in chat box. Kindly share your views on this session. And there's a surprise gift for you all. So what you have to do is to get the surprise gift is that you have to fill the feedback form, provide all the details, your email ID, your phone number, so that even we can have, if we have any opportunities, we can contact you back. And so don't forget to fill up the feedback form and get surprise gifts from Biotecnika. So thank you once again. Thank you all and take care.
Thank you.
Thank you all. Have a good night. Thank you, Dr. Prakash.